I'm CEO and co-founder of Matter Bio, and this is Sam Sharifi, my co-founder and CSO, and we'll be hearing from you as well. A little bit of background just to give you some flavor of what we're about to talk about. So we're going to share more about our startup, Matter Bio. It's here, it's New York based, and we're focusing on a very specific intervention for longevity and for disease prevention, we hope. And so our backgrounds are, I think, it's an interesting path to get here, so I just want to talk about that very briefly. So I have a background in neuroscience and cell biology and in computer science, and I'm an entrepreneur, a serial entrepreneur, so I had a company in healthcare that I was lucky enough to sell, and then I had the privilege to be able to ask, like, what's next? And what's the most ambitious, crazy, moonshotty, impactful thing I could possibly do with my life? And this is right around the time where this idea of longevity came around as like a real thing. And even back then, we're talking now about 6-7 years ago, I don't know about you guys, but I wouldn't really be able to tell people in any setting like what I'm actually trying to do. It was sort of like you could say it, but you gotta be really, time it right, maybe not at Thanksgiving, because, it sounded like crazy, just crazy stuff, right? But as we now are starting to see, what's cool is this is a scientific, evidence-based biotechnology type of approach and field. It's a new field. It's not just hocus pocus. So anyway, what we're really doing here, and we'll be able to share more detail, is that's our lens. We want to have biotech, biological, real interventions that'll be able to make an actual impact beyond all the important stuff that you all know about, like rest and nutrition and the usual exercise, right? So I'm getting chased by a fly here. So without further ado, so we're Matter Bio, and I'll just dig into it. Well, this will be short and sweet, so I'll give you a very high overview, and then Sam's going to get into some of the actual science. I'm just sort of, we got the actual scientist here, and then I think we're small enough group we can make it interactive, answer questions, talk through stuff. We also raised a round recently, so I was talking with Laura about it. But if any of you are also entrepreneurs or starting to embark on that journey or anything of that nature, we're happy to talk through our experiences at least and share more of that if that's of interest. Okay, so just to dig right in, let me see if I can, am I in the way? I'm kind of in the way, right? Here, I'm gonna do this. So we're used to thinking of your body as one body, one genome, right? You're born with the genes you've got, and every cell shares those genes. But that's actually not true. So actually, each of us is 1 trillion genomes. Every single cell has a slightly different version of your genome. And that's really the reality we're seeing. Every cell is different. And what do we mean by that? Well, why is that? Well, being alive damages DNA. It's a fact of life. Both exogenous and endogenous damage occurs. And what I mean by that is it's stuff on the outside of you that hurts it, and it's stuff on the inside of you that hurts it. So there's no way to prevent all damage. Even if you're in a perfectly sealed bubble, the processes that create life and create metabolism damage DNA. So it's inevitable. And these are just some examples of the types of damage. And so this is very difficult to avoid. You can't avoid it. And that's why we've evolved over millennia, all life has, ways of handling that damage. So most of the time it's repaired and you're fine. And it's kind of a miracle because it's damaged a lot, like millions of times a day. And most of the time it's repaired. A couple of times, I say a couple, but occasionally errors are introduced. The damage is not repaired all the way. And that's what we're really focused on. So those errors are basically 2 types. Point mutations, which are single changes in the sequence of your DNA, and then structural variants, which are basically a catch-all, which is larger changes. Taking a chunk and moving it to the wrong place, switching its order, replacing it with something else. These are all structural variants. So you can really think of it from an informatics perspective. These are noise that's introduced into the system, into the sequence of your DNA. Now this is where it gets interesting. So some of the technology to detect these point mutations and these changes is very new. And the reason is these are random. Every cell is different. And when every cell is different, actually being able to see them becomes difficult because if you didn't catch the right cell or you don't have a sensitive enough piece of equipment, you won't know whether what you're measuring is a mistake or an actual mutation. That's been the limitation so far. But recently, a couple of different groups have created technology to see these mutations, and they then ask the question, two questions. What do they look like across organisms, and how do they correlate with lifespan? It's a very simple question, and that's what we got here. So what this graph is showing you, this is from Alex Kagan's work on somatic mutation rates. They found two really interesting things. One, across all species tested, everything from humans to mice to giraffes, lions, dogs, all of us have around 5,000 mutations per cell at the end of life, roughly, between 2,000 and 5,000. That seems to be some weird upper bound. You don't see more than that. But even more interestingly, the speed at which you get those mutations is very, very tightly correlated with how long you live. That's what this is actually showing. So a mouse will get those 5,000 mutations in a couple of years. You and I will get it in roughly 80, give or take. And we suspect, although it's not in this graph, that some of the longer lived organisms like bow-headed whales and Greenland sharks and other types of these tortoises probably get it even slower, although that's not proven. I don't know if we have that data actually, but we would suspect that. And a really good example of that is the naked mole rat, which is a famous example of longevity in rodents. They're very similar to normal rodents, but they live up to 20 years longer. And so this is a naked mole rat. It's not the prettiest thing, but they drastically outlive their relatives by like really multiples of years. And in fact, if you see them, they're here on the graph, they actually get far fewer mutations on average per year than rats do. So this is all to say that these mutations seem to be very tightly correlated with lifespan, but we don't know yet if it's a correlation or causation. No one knows. So definitely correlated, but what does that mean? And I added this in because, as you're probably seeing, there's a lot more talk about going to space, going to Mars, going to low Earth orbit, spending more time up lower Earth atmosphere. Space is also really bad for your DNA.
In New York.
And it actually also accelerates aging.
Living here.
So this is also another kind of important converging piece of evidence that there's something about being up there that's really nasty. And we think, and we're actually hoping to show some of this data at some point, that it's actually DNA damage and somatic mutation increase is one of those drivers to be seen. Either way, we're going to have to do something about this if we're going to spend a lot of time in space or at Mars or who else, wherever else we're going to go. So a couple more slides on this. So some mutations are good, right? It's not easy enough to say all of them are bad. Some are beneficial. Most of them don't do anything. And some of them are really bad. And this is just some examples. You got clonal expansion, apoptosis, senescence. You can have tumors. Cancer is one of the clearest causes of mutation as far as most of our hypotheses show. So sort of multiple hit hypothesis of cancer is you get mutations in the wrong place, the cells gain advantages over their peers, and that's what we call a tumor. It's a very competitive group of cells. So we definitely know that some of them are bad. So what the heck are we doing here? Well, our goal is to take your genome back to baseline. We want to bring all those cells that are unhealthy, mutated, and divergent and bring them back to where they started. And the reason for this is simple. This is healthier than that. We don't know yet if it extends a second of life. No one's done this, so we can't tell you. But we know that this is not as good, generally, for age-related diseases and potentially longevity. So that's our goal. So then the question is, how the heck are you going to do that? Well, there's no magic way to do that. You can't, there's no one pill that's going to do this. It's a strategy. It's a convergence of approaches. So the way we're thinking about it is, you first need to understand mutation. You need to be able to quantify it, understand where it is, be able to read the genome. So you have to read the genome. You need to remove cells that are toxic. So you're not going to try to rescue cells that are too far gone, that have too many mutations, like cancer. You're not trying to get those cells back to normal. You just want to get rid of them. Clear the cells, clear the senescent cells, clear the cancer cells. And then for the ones that are left over, that are maybe on their way but not quite there, renew them. And what do I mean by renewal? Well, we're going to get into it. That's a very kind of hand-wavy term, but renewal is really taking that genome and figuring out how to revert it to baseline, actually. Yeah.
How do you stimulate genes to renew?
So that's a great question. So one of the problems with the DNA, once it's mutated, is your cells can't magically know what's the right sequence, right? You have two copies of each base pair, right? It's a pair. So those evolved to give you a backup copy of your genetics. But if both of those are corrupted, you're not going to magically, your cell doesn't know that that's a mistake. So we need to introduce information back to the genome. And we'll share in a second, again, high level, how we do that. But basically, that's that last step. And then you should be able to get back to baseline. So I'll stop there, and then we'll get into details. And Sam, I can do slides if you want. Or...
Okay,
go ahead.
yeah, so I will do some of this. So I'll talk a bit more on how we do it. So for each program, we have different technology to solve this. So for reading mutations, this cannot be done with normal next generation sequencing because we're looking for things that are in each cell different. So these are rare events. These mutations don't happen in every cell similarly. So we are using a new approach, an error-corrected sequencing called single molecule mutation sequencing, and with that we can sequence every mutation. We can find every mutation in the sample, and in addition to that, so it's known that that mutations go up with age, and there was a recent study that aside from epigenetic clock, they have developed a genetic clock where you can really see if you look at your mutations, what will be your age, and so on a genetic level. However, we still cannot do much about that, so we want to develop we are developing currently a editor to reverse somatic mutations. So with normal editing technologies, the problem is that they always go in and edit every cell, but these mutations don't happen in every cell. So what we are developing is kind of a spell checker. So it goes in, checks for this region, whether there is a mutation, and if there is a mutation, it flags it to the cell and reverse it. Now, we don't want to stay only focused on point mutations. Sometimes you have more mutations in one genome, so we have another editor that can do larger insertions, so we can insert DNA of up to 170 kb, so we can replace complete genome, so thereby we can slowly replace your genome or even optimize it. So think of like looking at variants that centenarians have and then putting those in normal humans and basically making from everybody centenarians, or even going one step further, looking at these long-lived species and looking, okay, what kind of proteins do they have? Can we bring those to humans and even live longer with that?
And Sam, real quick, so this is worth noting, this is brand new for us, that 170 kb is much larger than any other approach so far. And so this would be, if we can show this really, this is the first time you could in theory replace a whole gene. Before that, it's only been fractions of a gene. So it would be a big deal.
So to show you some data on that, so what we treated cells with puromycin. This is an antibiotic and it inhibits protein translation normally, so it's toxic for the cells. And normally, if you give it to normal cells, you see that they start dying. And if you have the resistance gene, then you can handle the antibiotic and the cells don't die. And so then we give the editor with this puromycin resistance gene and inserted it, and then we see that we get pretty good integration at different concentrations of this puromycin resistance in all three of our variants. We managed to actually integrate a larger piece of the genome with this editor in the cells. So for removing mutated or corrupted cells, we have developed a bacteria, Listeria attenuated, that is specifically being targeted to tumor cells. And with that, we can deliver a bunch of things. So we want, for our own internal program, we want to deliver tetanus antigens. So with that, if you bring that to the tumor, it will make the tetanus look like, make the tumor look like tetanus. And then if you're vaccinated against it, your body knows what to do. So they say, okay, this is tetanus, and clears it off. So the immune response is activated. So we decided to go for the most deadliest cancer to test this out. So we went for advanced stage 4 pancreatic cancer mice. And this is a very deadly cancer, and there's at the moment no proper cure. And when we give the treatment to the mice, what we see is a drastic decrease in the tumor size. So we can also see it on the pictures there. So here, this looks more like a healthy one, even though they have cancer. And what is more interesting is that even the metastasis, there was a drastic decrease of the metastasis after we treat them. So then we asked, okay, how is the survival of these mice? So we did the survival assay with them, and after their stage 4, we started treating them, and we see a 40% increase in survival, which is substantial for pancreatic cancer.
The equivalent would be, just to give it in human terms, like these are mice obviously, so it's days, and you can't, this is not how it works, right? But in theory, if we were seeing this in people, it could be 5-year difference. And pancreatic cancer is lethal within, most times, it's 5% after 18 months, I believe. Super rapid, super lethal. You usually find it at stage 4. It's nasty. So let's assume if we could find anything close to this in humans, this would be a huge deal just for this disease.
So this mechanism of the listeria going to the tumor should be tumor agnostic, so it should work in all solid tumors. And then we said, okay, let's test another very deadly cancer. So we went for advanced stage 4 ovarian cancer. So we tried the same in those mice. So basically we treated the mice at stage 4 again, and here we see even a better increase in survival, 54%. And then okay, we asked, so these mice are not curable because they're genetically engineered that they all of them develop cancer. So once the cancer is cleared, after some time they will get again cancer. So decided, okay, what if we treat them again? Will this help? So, and what we then see is a double in survival. So in principle, we could keep on treating somebody with cancer and keep on extending their survival a bit. And so this is our team. So we have Alex Meslov and Jan Feig from the Albert Einstein College of Medicine, who are somatic mutation experts and inventors of the sequencing approach. George Church from Harvard, who's the gene editing expert. Claudia Gravenkamp, who's also at Einstein and is an expert in listeria. And then we have Desso and Yunan, who are our scientists who are pushing the programs in our lab.
So I think maybe, and that's all we've got. We could go on for hours, obviously, but that's it for this.
Any
Sorry.
questions?
Yeah, we guys